Okay, we're going to pick up here. I've one additional note to make. I forgot to mention it before we left off with the last part. But the sound, whenever we're hearing, and we have this sound wave. Or initially, we had a pressure wave, right, and then it is transmitted to the vestibular duct, moves the basilar membrane, moves the hair cells against the tectorial membrane, right? And we get this action potential. Once we get this actual action potential that we discussed, it's going to travel along axons of neurons, and these are going to merge and form something known as the cochlear nerve. Okay. Now, the cochlear nerve is one part of a larger nerve known as the vestibulocochlear nerve, and that's what we see right here. This is our eighth cranial nerve, and this has to do with sending information to the brain regarding hearing, so audition, along with what we're going to talk about next regarding equilibrium and balance. Okay, so there can be information be sent to a region known as the superior olivary nucleus of our brainstem. We can also have a region known as the inferior colliculus receive information, and this inferior colliculus is involved with auditory reflexes. So whenever you hear a loud noise, or you hear somebody say your name, or some other auditory stimulus, and you turn and you orient yourself to that, this inferior colliculus is coordinating that. And then ultimately we go to that medial geniculate body of the thalamus, and then ultimately to the temporal lobe where we have our primary auditory cortex. Okay, so I just wanted to make that clear before we move on talking about equilibrium. Okay, so equilibrium, this has to do with our awareness of where we are in space, the position of our head, and our overall balance, right? And so we have different types of balance. We have balance and the position of our awareness in an angular setting, so in angular rotation. We also have it in horizontal movements as well. Okay, so directly side to side, directly up and down. Okay, whereas angular would be something like spinning. Okay, so this is monitored by something known as the vestibular apparatus, and we'll talk about a few more features of the internal ear that will be specific to our sense of equilibrium. All right, so the information from all of this is going to ultimately be put into a nerve known as the vestibular nerve, and this is the second part of the vestibulocochlear nerve. So we mentioned just previously the cochlear part. The second part is going to be the vestibular part that is forming this eighth cranial nerve. All right, so we'll talk about a few regions. Some of these are going to be known as the vestibule. We're going to talk about things called the utricle, the saccule, and we're also going to talk about the semicircular ducts and the semicircular canals. All right, so this idea of balance, balance is a multifaceted thing. So it has to do with our sense of vision, right? So whenever we see, it has to do with our actual vestibular apparatus, so the actual sense of balance. It has to do with proprioception, so having to do with our awareness of joints and muscles and tendons and ligaments where they are in three-dimensional space. And kind of all of these things together help to coordinate our overall balance and our overall equilibrium. So what we're going to look at first is we're going to look at static equilibrium, okay, and linear acceleration. So in other words, when we're not spinning and we're not rotating or anything like this, okay, we have our head more or less still, and we move our body, or our head tilts down, or our head tilts up. Okay, so this is happening, and this is occurring in a region known as the vestibule. And the vestibule, when we look in our inner ear, the vestibule is this general area right in here. This is the vestibule. All right. Within the vestibule, we have these regions known as the utricle and the saccule. Okay, and these are going to monitor, and these are going to be responsible for detecting linear movements and linear acceleration. So some are going to be sensitive to changes in vertical movement. So this is specifically going to be the saccule, and then the utricle is going to be sensitive to horizontal movement. So this will differ depending on whether we're moving side to side or whether we're moving up and down. All right. So in this vestibule, we have this kind of thick gelatinous fluid. Okay. With this fluid sitting on top of it are something known as otoliths, and otoliths are essentially these little calcium carbonate stones, or these little crystals that sit on top of this gelatinous fluid, and these stones will move over the top of the fluid will distend and distort and move and pull these little hair cells, similar to the hair cells we talked about whenever we talked about hearing. And the movement of these hair cells can generate an action potential, or, depolarize action potential, and then send this signal back to the brain, right? So as we move our head in our ear, we're going to move these little crystals, these little otoliths. And when these otoliths move, they're going to move and pull on this gelatinous material. And when this happens, in the direction of gravity, obviously. And when this happens, we're going to distend those stereocilia, similar to the stereocilia we talked about during our hearing lecture. And it's going to distort those cilia, generate a depolarization, and then an action potential, and this can be sent along to the brain. So we actually do have little rocks, these little rocks in our head. Okay. So whenever we're looking here, we'll have different types of movement produce different types of distortion and distension of these hair cells, depending on the movement of the otoliths and this general kind of gelatinous material. Okay, so this is known as the macula here, this region here, and you have a utricle of a macula, and you have the macula of the utricle and the macula of the saccule. And again, depending on if we're moving horizontally or vertically, we'll be talking about one or the other. Okay, but the idea is basically the same. So as we tilt our head, right? So imagine we're tilting our head down this way. So too are these otoliths going to move in that same direction. They're going to pull on this gelatinous material, or they're going to distend these hair fibers to move in that one direction. Okay, and again, similar to hearing, whenever we distend and we pull those hair cells, they can open. And when they open, they can allow ions to flow in, and they can result in depolarization and ultimately the generation of an action potential. Okay, so it depends, like we said, on the specific movement. Are we moving vertically or are we moving horizontally? All right. So here we can see different the different macula of the utricle and the macula of the saccule. So here's the saccule. So we said the saccule is responding to vertical movement, right? So here we have vertical, right? How it is oriented vertically. So movement in the vertical direction would move these hair cells, whereas the utricle is detecting horizontal movement, right? So any movement in the horizontal plane is going to distort and move these otoliths. It's going to move this gelatinous material one way, and then it's going to bend those hair cells in that same direction. You can generate an action potential. It can be sent along this axon of the neuron to ultimately make the vestibular nerve, and then this can be sent to the brain. Okay, so this all has to do with linear movement, right? We're moving in a linear direction. So, are we moving vertically? Are we moving horizontally? Okay, so here's another example that is showing the movement of these otoliths over the gelatinous material over the cilia. And again, we have the stereocilia and we have the kinocilium, just like we had with our sense of hearing, right? So the basic process is a little bit different, but the end result, with the movement of the stereocilia, the movement of the kinocilium, that's basically the same, right? So again, as we move toward the kinocilium, we're moving toward that largest one. We can open up ion channels, we can depolarize the cell, we can release neurotransmitters, and then we can get a signal being sent along the vestibular nerve, right? Likewise, if we move away from the kinocilium, we can close those little channels, we can hyperpolarize the cell, we can release less neurotransmitter or no neurotransmitters, and we will not have action potentials, or we'll have fewer action potentials being sent along the vestibular nerve. Okay, so it all depends on the specific type of movement, and it depends on the specific direction of the movement as well. If we're moving toward that kinocilium or we're moving away from the kinocilium. All right. So that was the macula of the utricle and the saccule. And again, the macula, I don't know if I described it very well, but this is where the hair cells of the utricle and the saccula are kind of clustered together in this kind of oval-like structure. This is known as the macula. So you have a macula for the utricle, and you have the macula of the saccule as well. And again, those are detecting linear movements. They're detecting movement in the vertical direction, the vertical plane, or the horizontal plane. So the second kind of part of our vestibular apparatus has to do with our semicircular canals, all right? And these semicircular canals and these semicircular ducts are going to be involved in angular movement or angular acceleration. So movement in an angular plane, right? So instead of moving the head directly this way or directly this way, we may be rotating it around in a combination of directions, okay? And so we have three different semicircular ducts and semicircular canals that detect movement of the head in one of these directions, right? And so we have an anterior, we have a lateral, and we have a posterior. So whenever we shake our head side to side to say no, this is going to be detected by our lateral semicircular canal. When we move our head up and down like we're going to nod yes, this is going to be the anterior. And then if we tilt our head to the side, so imagine you bring your right ear to your right shoulder, that's going to be the posterior duct or the posterior semicircular canal. All right. So each of these are working independently, but they're also kind of working together to detect angular movements of our head. Okay. So we have a region known as the ampulla. Okay, sometimes known as the cristae ampullaris. All right. This is this kind of whole region that we find in this area here. This is known as the ampulla. This large kind of gelatinous structure that you see here, this is known as the cupula. Okay, the cupula is this kind of gelatinous blob of material that is sitting over the top of our ampulla here. It's right here, and there are also hair cells that we have embedded in this area, right? So just like we had with hearing, just like we had for linear movement as well, we have these for angular movement as well. As we rotate the head, so as we are spinning, as we're rotating the head in a particular direction, we are going to move the fluid in this area, and again, the fluid here is called endolymph. The movement of the head is going to move the direction, or is going to cause the cupula, and cause the endolymph to move, which is going to deflect the cupula. And as you deflect the cupula, as you move it in one direction, you're going to bend the hair cells, bend those stereocilia in that in a certain direction. That's going to result in the stimulation of an action or depolarization, and then the stimulation of an action potential that will then be sent back to the brain. Right. So here we have, as our head rotates, we have the fluid surrounding these cell bags or these cupula here. The cell is going to lag behind. It's going to move in the opposite direction of the head movement. So imagine if we moved our head quickly to the right, or if we moved our body quickly to the right, the head is going to kind of lag behind. And so you see that same thing happening. And you then get distension and pulling of those stereocilia, and you get ultimately the generation of an action potential. Okay, so we're going to stop here with our sense of equilibrium. So it's a little bit shorter than our sense of hearing. And then we'll pick up with olfaction, and then we'll end with our sense of taste.